I'm really happy to be here in the Dataton Africa. I'm Clara Arboleda. I'm working right now in the German Center for Integrative Biodiversity Research in a project that is led by Stephanie Jorbok. But today I'm going to present a work that I'm working on for the last 2 years that is called Exploring Biogeographic Patterns of Bacterioplankton in the American Freshwater Insight from the µSudAqua database. This is a work that belongs from other network that is from South America. I'm from Colombia. And I want to talk about a little this network because can inspire the researchers on Africa, maybe have similar ideas or maybe can get some insights of this. So the µSudAqua network is a collaborative network in microbial aquatic ecology of Latin America. The main goal is to build networks of researchers focused on microbial aquatic ecology, create collaborative environment that promotes long-term research partnership and also training opportunities. This is an idea of 3 researchers from Uruguay, Brazil, and Argentina. That is Cecilia Alonso, Hugo Sarmiento, and Fernando Unrein. They, at the beginning, start these 3 researchers, but right now we are like more than 100 people working on this. And now the new coordinators of these networks are Paula Huber, Micaela de Melo, and Juan Pablo Niño. And as you can see, all of them belongs for different countries from South America. This network has different working groups. These are the 3 main working groups. There are the microobservatories for South America, patterns of South American aquatic microbiome, also morphological and functional traits. And all of these main groups works in different environments and ecosystems across Latin America. As I told you, this network already has more than 100 participants and 26 research groups and different Latin American countries. You can enter here in the link and you can see more about this network. So today I'm presenting a work that was also presented in the ISME this year. This is an effort, it's not only my effort, it's an effort of a lot of people of this network, principally with Juan Pablo Niño, Natalie, Carolina, and Hugo. So I'm going to start and show you a little of the results that we got already. So we know that Earth hosts an amazing diversity of life forms. Biogeography focuses on the study of the geographic distribution of these organisms and the patterns observed in species diversity across space and time. And as we know that because we are a network from microbiologists, microbes are the organisms that are more abundant in the world, and also they play crucial roles in biochemical processes and everything. So right now, with the development of next generation sequencing, we can move from study local scales, I mean environments that are near to your home or just your country. But with these tools, we can now study bigger scales, like regional scale or global scales. That, for example, the invitation with this dataton is like to have more data from Africa to increase these scales and can have the opportunity to ask different questions that we were asking right now. So biogeography can be studied with a focus on ecological factors and also historical factors that have shaped and continue to shape the distribution of organisms. The ecological factors are environmental factors or also biological interactions. But with the historical factors, I mean the 4 main processes that are speciation, drift, selection, and dispersal. And all of these, as I said, shape the distribution of the organisms. So right now, as Stephanie told us at the beginning, and also Pedro and also Emma, despite the abundance of data, we still have significant geographical gaps in understanding microbial diversity. And these gaps are mainly in the global south. We can see this in this paper that I'm going to explain better later. that is the µSudAqua database that show us that we have less points of microbial diversity in the south, and also the mycoda database that we are going to talk about this database through the 3 days of the datatons. So we can see that the gaps are mainly in the south. Yeah. So how can we study the biogeographic patterns of bacterioplankton in the southern hemisphere? So one method that we use is, first of all, we pick up database as a control. This is a database from Canada that is Carbax. Each of the colors show different ecoregions. There are regions that has different climates and also forests, that this can shape the microbial communities, or is a hypothesis that this can select some microbial communities. And also we took the information from one paper that is a product of this network, that is µSudAqua network. That is a data paper with a georeference amplicon data of aquatic microbiomes from South America. This database has more than 800 data from the south. From this study, we took only 178 lakes from the south, also for different ecoregion, as you can see for the colors. And in Canada, we took the same numbers of lakes. So all these data were processed by the same pipeline of DataDOS. So we got ASVs that are like our proxy of a species. And we got more than 50,000 ASVs in this database. So at the beginning, we start to ask questions more on a community level, also related with the ecological and historical factors. So for the environmental factors, as I told you, we have different ecoregions, that this is already like a proxy of the environments where these microbial communities are. So the first question that we had is like, if these communities are similar or not. So we made an MDS. I don't know the level of the people here, so I'm going to explain how you read these graphs. All of these points is a microbial community. The colors belongs to the ecoregions that you can see the distribution in the map. So if these points are more closer, it means they are more similar to the community, and if not, they are not similar. And as you can see, we have a very important beta diversity here. The composition and similarity of this microbial community differ among ecoregions. So this reflects environmental preference. That means that in these different parts of the map, we can find different microbial communities. This is on a scale of ecoregions, but with this big data, and that is the idea of the dataton, to increase like the scales of the data. If we increase the scale, for example, this is a scale of climate zones. We have here the boreal zone, the tropical zone, and the austral zone. So if we compare the microbial communities that are there, we also see that there is a similarity between these communities. So also they are reflecting a preference of these climate zones. So this is an example of how the scale can tell us more about the microbial communities. And and that's the idea, like to work with these big data. For historical factors, I remind you that are the 4 main processes that are selection, dispersal, and drift. There is a methodology that is well used, that a lot of researchers already use, that is the Stegen approach. You can find it here, the paper. And this approach takes into account the phylogeny of the communities and the turnovers between the samples. And with this approach calculate the percentage of homogeneous selection, heterogeneous selection, homogenizing dispersal, dispersal limitation, and also drift. So we analyze the percentage of these processes between both databases, the one from µSudAqua from the south and the other from Canada, the Carbas, and also inside the µSudAqua database between ecoregions and within the ecoregions. And as you can see, when we compare two databases, one from the South and one from the Canada, the dispersal limitation is bigger, and that has sense because we have communities that are separate. So yeah, we just have the South of the South America, so we don't have the center of this center of America, so we can have this dispersal limitation. But when we go inside the µSudAqua database, we saw that the limitation of dispersal decrease, but increases the selection. Yeah, and as we saw in the last graph here, I want to come back. We already saw that the environment selects these communities, so have sense that the selection increase when we see each of these data. As I told you at the beginning, there are main four processes: dispersal, drift, and selection, and speciation. But with this approach, we can now analyze the speciation, like new species. So we take a proxy of novelty of new species in this community. That is this index. There is the microbiome novelty score. I put there the article if you want to check out. This index, what it does is compare each community that you have with a global database that is the cheetah. And this gives you an index of how novel this community could be comparing with this global database. This is very cool because we already checked that the µSudAqua database are above this threshold, the red line. So that means that the sample from the µSudAqua database are more novel comparing to this global database. And this global database are all the samples that are available. So this could be also very interesting for the African researchers because this new sample that is going to enter and that we are going to gather can bring like more information from novel communities of microbes that we don't know and also are missing in these global datasets. Also in this graph, I want to just point out that the CARBAS, that are our control database from the north, from the Canada, is below this threshold. That means that these communities are already very common in this global database, so they are not very novel. So that also shows us that these communities from the South are increasing the novelty in bigger data sets. So at this moment, we have an analysis of a community level, but a question remains, is which components of the community are associated with these processes. This is not very easy, and we're working still on that, but the idea is to divide the community into group of ASBs that exhibit current ecological behaviors. This is not very obvious and take us a lot of time, but I think that we are in a good path. So what we did is just to do the same that the people from microorganisms does, like they just put the organisms in the map and see their distribution, and then start to answer a question related to the distribution and some hypotheses of the processes that are occurring and that can describe like this distribution. So for that, remember that in this work, we have different ecoregions that have these different colors. So what we did is just make a map. Then we put the sample from the south, the more south samples until north, that is in Canada. And then we see the preference of these organisms by ecoregions, but also by a bigger scale, that is the climate zone, austral, tropical, and boreal. And also for those that doesn't have any specificity or selectivity by habitat, yeah, because for this, we run this approach that is the INVAL. This is an index that you can run and give you how one species or an ASV prefer an habitat. So you can detect some habitat specialists that prefer some habitat, or some of these ASVs that doesn't prefer anything because they are very broad distributed. So here in this map, we have 3 panels. In this one, we have the environmental preference in a bigger scale, that is the climate scale, austral, tropical, and boreal, then the environmental preference by ecoregions, and then those that are no specific or doesn't have any selectivity. So this is just a map that the idea is to create groups of ASVs that have the same pattern and maybe answer for the ecological processes that we already talked about it. So here, first, we divided the community in those ASVs that are in both database, in the µSudAqua database and in Carbax. So you can see that these panels are with color, the entire panel. So that means that the ASVs are in the whole, in all the ecoregions. So these are ASV that has a broad distribution, are cosmopolitan. They are in all the lakes in the south and in the north. Some of them has an austral, tropical, and boreal preference, but some of them doesn't have any preference, so they are just in all these aquatic environments. And this show us something very important. It's like there is no dispersal limitation in a continental scale. Yeah, and that is nice because that means that we can find these groups across like the whole continent. So we have other groups that also they are broad distributed in the map, in these samples that we have, but they have a much more patchy distributions. They are in most of the ecoregions, but in some of these ecoregions they are missing. So that means that they have no dispersion limitation in a continental scale, but the selection is working harder for them. So in some environments, it seems that they cannot survive, but it doesn't mean that they are not like coming or going, because they can, like, they are appearing in different ecoregions that are close by. The second group are ASVs that has a dispersal limitation in a continental scale. That means that they are only in the µSudAqua database or in the other one. But in each of these databases, they are broad distributed. So that means that they have this limitation from the north and the south, but they show no dispersal limitation in the regional scales. So as you can see, there are like different processes, ecological processes that are working, but in different scales, and that is great. Because maybe that kind of patterns you cannot see if you have a database that is shorter than one that is bigger. That is the idea of the dataton that we are working during these 3 days. And the last groups are these one that has more a narrow distribution. They are just only in one ecoregion, in 2 ecoregions. That means that the selection and the dispersal limitation are working harder with this group. So as I resume, we put these 50,000 ASVs. We can organize them in just 8 groups that exhibit a coherent ecological behavior. And some of them has this continental scale. They are broadly dispersed. They have no dispersal limitation between the continent, but some of them has more deterministic processes that are operating. And the others are more regional. Yeah, they are not in both a database, and also there are different processes that are working on. Right now we are working on the hypotheses of these groups and explaining better and doing some statistical tests to check if this is not like a random distribution, and also we got like a clear patterns. But something that I want to highlight is also we study if these ASVs are core, like they have a lot of occupancy and abundance, or they are accidental or rare species. We found both, but with these 2 both, we can see that they have a biogeographic patterns too. That is very important, because some of the researchers just eliminate these rare species for the studies, but here we are showing that they also have a biogeographic patterns. So right now also we are working in the taxonomy. We are putting the names of these groups and looking who are these ones that are cosmopolitan across freshwater environment, and also which are rare, which phyla are more important than others, etc. So the highlight at a community level is that we found a greater novelty in the South American samples compared to our external database and a global database. Remember that the external database is that one from Canada and the global database is Cheetah. And this is really important. I want to highlight this from the African researchers, but because putting this new data available can increase the novelty of this South America, this global South microbial community, and maybe we're going to have more information. And the other highlight from the community level is that the ecological processes are dominated by dispersal limitations and homogeneous selections. But at population level, we can, with this methodology that is doing the same thing that the microecologist does, the people who study microorganisms, we saw what the patterns of these microbes would tell us, and we can divide these ASVs into groups that have a coherent ecological behavior. This also can give us some information about the ecological patterns, dispersal limitation, also selection. Also, we found that some core ASVs that we always say that are the most abundant, that has a higher occupancy, we saw that they don't necessarily respond to the same ecological processes. There are some that are really cosmopolitan, but some of them has more pressure about selection processes. And finally, also these rare species or the accidental species they are not so accidental. We can see that they have some distributions and some ecological patterns that we can put it in the map and tell us more information about them. So I want to thank Juan Pablo, Natalie, Carolina, and Hugo, and all the people that help us a lot, the people who made the database, also who help us with different analyses. As you can see, we have different approaches. This work is already divided into works because it has a lot, but we are close to close one of them. And also, I want to add that this µSudAqua network, that is an example of how researchers from a regional scale joined to make questions with bigger databases that could be an inspiration for African researchers, because as we hear by Pedro already made a network from soil. Pima has with more marine environments. Maybe it's missing, or maybe I don't know that it already exists, a network from freshwater ecosystems. And I want to tell you that this regional network is already joined this global network that is led by Stephanie Jorbog. And this is the network that is inviting us and create this dataton. And these samples are already in the MyCoda database, in the Dataton of Latin America that was in 2023. And the idea with these datatons is to also increase more the data here in the map from Africa and maybe start to do some questions. Yeah, I want to thank also the African Dataton staff that right now we are working in these 3 Datatons days, and I think that has been really amazing, and I've learned a lot. And yeah, I think that's all. Thank you.